So my primary research focus currently is we work with feed efficiency in livestock. And so feed efficiency is a really big topic right now in the livestock industry. Producers want to know how they can maintain production while feeding animals less, or in other words, we could also improve stocking rates. So there's been a lot of drive to research that area. And so our primary goal is to help producers find tools to measure feed efficiency. It's really hard to measure it because it's time intensive. It usually requires about a 70-day test. They also need to have specialized equipment to measure individual feed intake on group housed animals. And so that's why we're trying to find tools in order for producers to be able to better do this. It's hard to do, it's difficult to do, and expensive to do. So if we can find things like genetic markers in order for producers just to take a blood sample, send it in, be able to tell which animals are going to be more feed efficient than others. Some of the current thrust of our research is looking at the rumen microbes. Basically, the ruminant animal is a host to the microbes that live within its rumen. And there's been a lot of work into looking at how those rumen microbes may be controlling how feed is broken down and how efficiently. And so our goal is to maybe use something like a panel of rumen microbes that are indicative of an animal's feed efficiency. So we look at the microbes on a DNA level in order to identify them, and ultimately also look at maybe some of the expression differences there. We use both cattle and sheep. And so basically I work a lot with sheep, not only because they're very important to the Wyoming livestock industry, but also because we can use them to kind of see how cattle might react as well, and vice versa. We use a lot of cattle studies to predict what might happen in sheep. There are some genetic marker tests out there for feed efficiency in beef cattle, not so far in sheep. That's why we're working on that line of research. The problem is that once you have genetic markers, sometimes they're really consistent, but sometimes they're really not consistent. So we still have to keep refining any genetic markers that we do have, because there may be differences across breeds, across different environments, across different feeds. And so while this set of markers might work for one set of producers, it's likely not going to be universally true for all producers. But I think that we'll keep refining those, and we'll open up a whole new line of probably markers ultimately for feed efficiency. Basically, if you were a producer and you could send in a sample, and especially if you're choosing maybe which females to keep back as replacements, or potentially maybe which bulls or which rams you were going to use for your breeding stock, then you could have a better indication of they're going to be more feed efficient, their progeny are going to be more feed efficient, and your whole flock or herd is going to be more efficient as well. Maybe you don't want to use it on breeding stock, but maybe you want to use it to select those animals that are going to do better in the feedlot and send those off. Basically, we're giving a producer more confidence in the selection decisions that they're making. We know that it's a prediction and that there's going to be some cases in which it doesn't hold up as well. But again, we're trying to account for more of that genetic variation. The more that we account for that, environment's still going to play a big role, but at least we're accounting for what we can. So another area of research that we've been working on, and we've been trying to tackle this area of sulfate toxicity, and I've been going at it from more of the genetic standpoint. And by the genetic standpoint, I mean, are there different molecular processes that are making an animal more susceptible or more tolerant to the higher levels of sulfate? Because what we know is that some animals can have a lot of sulfate and they're fine. Some animals can have a little bit of sulfate and they don't do so good. And so we know that there's some inherent part of an animal that makes it more or less tolerant to these high levels of sulfate, and it probably goes for a lot of the other dietary conditions as well. So one thing that one of my current students is doing is she has recently done a lamb trial where she gave lambs high levels of sulfur water. That's something that we really encounter a lot of in Wyoming, is a little bit of higher sulfur waters. And then what she's doing is she's looking at it now from the rumen microbial standpoint. So that's kind of where my two areas of research have been coming together recently. She took rumen samples from those animals. We identified ones that were more tolerant to the high levels of sulfur in the water and ones that were less tolerant to sulfur in the water. And what she's doing is extracting the DNA from those rumen samples, and then she's looking at it in terms of which microbes are maybe in higher concentration in those animals that are more tolerant to the high sulfur water. Is there ways that we can develop treatments off of that? Animals that we know that are going to go into high sulfur situations, maybe we can somehow change their diet or give them a supplement in order to change their rumen microbial population to allow them to be more tolerant to the high levels of sulfur. As researchers, it's important for us to remember that down the road we need to answer questions that are important for producers if we're going to be researchers in animal science. And while we study a lot of molecular mechanisms and things at the DNA level, we're looking at how those things control what happens to the whole animal. And so we always need to keep that big picture in mind. So I think as long as we listen to producers, listen to what's important to them, I think we also have to listen to consumers, what's important to them. And I think as long as we keep listening to them, that will help us to drive our research in the right direction.